afternoon, everybody. Good morning, of course, Simone. Simone, thank you very much for being here today for the second part of your seminar. I guess that today you will be talking about different statistics compared to the t-test that you showed the last Thursday. So I think you can simply start with your presentation. So as usual, you've got the chat that you can use for questions, and at the end of the talk, Simone will gladly take questions from the audience. So please go ahead, Simone, and start with your presentation. I'll give you the host.
Thank you, Andrea, again for the invitation, for coming back. I'm grateful that I had two opportunities to to finish my story. Let me share the screen here. Here we go. Perfect. So today I want to continue the episode from last time. Will take about half an hour, and I think I hope that at the end of this 30 minutes presentation, you will feel more comfortable with dealing with proteomic table if you are a non-specialist. Again, these lectures are not meant for people that are already very experienced with proteomics. They are more directed to people who are using proteomics, but they are still a little insecure with what they do once they obtain a table of interesting proteomic data. So I call it on purpose this lecture Unbiased Hypothesis Generation, because I'll try to show you how we use a proteomic table to tell us what is interesting in there, rather than the other way around, rather than go hunting for our favorite proteins. So we were here at the end of the previous lecture. We discussed how you convert a raw proteomic table into a table sorted by a significantly enriched feature. And these are, you remember that we discussed how to look for enrichment generating a fold change, how to study the confidence of how reproducible your observation is, calculating a p-value. And then we created this arbitrary score to help us sort the table for most interesting proteins, proteins that have the strongest fold change and p-value, to get them at the top of the table to make it easier, our visualization. But still, in the end of a proteomic experiment, you might end up with tables of 5, 6, 7,000 proteins and maybe 1,000 regulated features. And then the question is, okay, so what is interesting in here? Potentially everything. In practice, I need to start from somewhere, without being too much overwhelmed. So just a reminder, consider this. In a simple proteomic experiment, let's say you have two conditions. Essentially, you have three quantitative dimensions that you can exploit, depending on what you want to focus on. Now, the first one, as we discussed, is the fold change. So one column in your table tells you what is enriched in one condition or the other condition. The second one is the p-value, which doesn't tell you what is most enriched. It more tells you what is the chance that this experiment is going to be the same, if you do it again and again and again. So it tells you about the reproducibility, it tells you about the confidence of your observation. And then you have a third dimension, especially if you work with label-free data. The intensity of the protein itself, no matter how it has been calculated, is still an interesting observation and potentially useful if you want to focus on proteins that are more abundant. Right, so abundance and fold change are two different things. Fold change is how much is enriched in a protein in condition A compared to B. Abundance tells you how much protein you have in your sample. And this could be very helpful, for example, in a volcano that has thousands and thousands of data points. If you, for example, scale the bubble size, this you can do it easily in Excel, by protein abundance, you can get a feeling about, okay, there is a lot of regulated proteins out here, but can I focus on the one where I have more into my sample? And why this can be useful? This can be useful for the principle that A protein that changes from 10 copies to 20 copies as a twofold change in enrichment, but also a protein that changes from 1 million copies to 2 million copies as a twofold change in enrichment. But potentially the ones that you have 2 million copies of it in your cell could have a more higher impact on the biology of your cell. For example, when you're looking for biomarkers, if you look at this figure here on the right, when you're looking for biomarkers, it's important to find a protein that is very specific for your sample, but it also helps a lot to find a protein that is not only specific, but is also highly expressed. And that's for multiple reasons, not the last one, the possibility of producing an assay to hunt for this protein that doesn't require a lot of sensitivity. So remember that, in a simple proteomic experiment, you have these 3 quantitative dimensions that you can use. And now, let's see how we exploit them, right? So the first question that you have when you have these big tables is, what is the meaning of my regulated proteins? What are my regulated proteins? And usually the thing that people refer to at first is gene ontology enrichment. Gene ontology enrichment, gene ontology is a database that includes a lot of information about protein functional properties, protein where they are localized in the cell, what is their molecular activity, and also what is their impact in biology. It's very, I'm not going to say that it's not useful, of course, because lots of people use it and people extract a lot of important information for it. It tends to be a little underwhelming. It tends to be the type of annotations that you find specific for your protein of interest. They usually end up being a little bit generic, and they don't always tell you what is really going on with your sample. So it's a very useful step, but it tends to be a preliminary step. And then the other important question is, how do I study the functional enrichment of my proteins in this big table? What do I do? Do I take the top 100 proteins in the top of my table? Do I take my entire list? Do I take the proteins that are significantly enriched? If you take those that are significantly enriched, you need to choose a hardcore threshold where you filter for a specific p-value, for example. You can do it, of course, but always remember that p-values are, they are detectives. They are not judges, right? So they are not numbers they are indicative numbers that suggest, what is the probability that this is going to happen again. They are not the ones to decide the destiny of your sample. So the question remains, what proteins I take and what kind of software I use to look for this functional enrichment. Now I'm showing you here David. David is one of the most famous. You just click on functional annotation, you paste your genes of interest, and then it tells you if they are specific for a specific, function. There are others. There are actually many, many others. I feel a bit guilty only showing a couple, but I'm just showing you the ones that we use the most. Gorilla is another one of them that performs gene ontology enrichment. I'm showing you this one on purpose because this is one of those that is the easiest to use, and it takes into account the fact that you need to enrich something compared to something else. Here is what I mean. So basically, when you upload your list of genes or your list of accession numbers in your proteome in a software like Gorilla, this one is asking you, do you want to paste this list of genes as a single ranked list or a 2 unranked list? So this is actually very important. What's the difference between these two? Like single rank means that the order that you paste your list of proteins is going to be used to define what is enriched. So in other words, the software is going to tell you what is enriched at the top of your table compared to the bottom of your table. If you paste it as a two unranked list, you need a target set that is going to be your proteins of interest, and a background set. And the background set is going to be, the rest of your proteins that you have identified. When software that don't ask you anything, usually they use as a background set, they use the entire human proteome or whatever organism you're working on. And this is dangerous. You have to be careful because ultimately you want to know which proteins are regulated by my drug treatment, right? But if you are doing this experiment with myotubes, for example, both your upregulated proteins and your downregulated proteins are probably proteins that are involved in muscle cells. There's probably myosin everywhere, regulated or not. Especially if you are not very sensitive with your experiment, you're always going to have a bias of proteins that you can see in your model system. So in that sense, whatever proteins you paste in this software, if they are the upregulated or the downregulated or the non-regulated, functional enrichment is probably going to provide you muscle fibers, activity in muscles, right? And this is not, this is misleading you because it's not telling you what is specifically upregulated in your data, compared to the rest of the proteins that you detect. So I'm going to show you a practical example at the end of this presentation. Just keep this in mind. Right. So, and then just as a reminder, there are many other databases and software that you can use to look for functional enrichment. You can enrich for pathways, you can enrich for diseases, you can enrich for protein families. This is just two examples. This is KEGG here on the left, and there are many software that use the KEGG database to look for pathway enrichment. Here on the right is GSEA, developed by the Broad Institute in Boston, and then this one instead is a software to look at what is functionally enriched in terms of, well, a lot of properties, but is mostly focused on patterns that are reconciled to specific diseases. So depending on what is your question might not be what is the molecular function of my enriched proteins, could be what pathway they belong. You can use a software that is more, specific for that specific question. But ultimately, what you want, something that is digestible to the human eye. So let's say that you find that one of your enriched functions are proteins related to mRNA processing. Okay, now your next question is, what are the proteins in my list that are related to mRNA processing? So you ultimately want to have data that are, digestible by the human eye, so they look sort of like this. And we get there in a minute. And now the real question is, how do you go from a functional enrichment to a list of proteins communicating with each other somehow that I can visualize, and it also becomes, appealing for a scientific publication. Well, the thing is, there are many databases, again, available online that can be helpful to connect proteins to each other. Right? So when you do a proteomic experiment, you get out with a list of proteins that are enriched, but the real question is, do these proteins have anything to do with each other? So your single experiment does not give you that answer, but what you can use the immense literature that we have to at least start clustering proteins in groups, grouping them with each other. And again, there is a number of databases that can do that because these databases contain information about protein-protein interaction. In some cases, there are physical interactions, for example, like Bioplex, developed by the lab of Stephen Gigi. This is a database that includes thousands of immunoprecipitation experiments of each individual protein, and then it contains what kind of information about what kind of interactors your specific protein has. So you can use it to build a network of protein interactions, theoretical, of course. And then there are other databases, for example, like String, and String instead contains a lot more information than physical interactions. It contains, for example, associate proteins depending on text mining, depending on co-expression. there are different ways to basically create a link between proteins that are found in your database. How they work, I'm going to show you actually in a minute with a practical example. But essentially, those databases, most of them, you just add your protein of interest, you click search, and then it turns out a table, a diagram that looks a little bit like this, depending on what you input. And this type of clusters, I mean, from my perspective, what I would do, I would not look at them, protein by protein, especially because they can be very big. But what they can be helpful for, they can be helpful, for example, to help you identify, like I said, like cluster of proteins. There is an entire group of proteins that are all involved in this specific interaction. I would maybe find it more interesting to start from that cluster rather than starting from a single protein that seems to be involved in another function, but it doesn't have any other proteins related to that one that is co-regulated. Of course, it's an arbitrary decision, but consider that it's very, very rare that in a cell, proteins act alone. So, identifying protein complexes or protein clusters could be very helpful to gain confidence that this is really happening in my proteomic experiment. These software, like for example, String, they also have analysis patterns that you can color proteins for a specific function that helps you tell, okay, what are all the proteins involved in this or that. Obviously, here keep in mind that, like I said before, String, for example, uses as a background the human genome. So these p-values that tell you how enriched is your function, that they are biased because your experiment is biased. When you analyze any kind of cell, you're not going to see all proteins. You're only going to see proteins that are more specific for that cell type. Yeah, and then string data can be exported. You can upload them in free software, for example, like Cytoscape, where you can start playing with the aesthetics of your network. Cytoscape, to the best of my knowledge, is the best free software to help you organize your data into networks. We don't have time today to discuss how to use Cytoscape, and I'm sure that there are other videos where they explain it better than me. But just to let you know, this is a great resource to create some very nice graphs. All the ones I show you so far were done in Cytoscape. Other options regarding how can you tell whether in your list of regulated proteins there is a set of proteins that probably have something to do with each other. So there is probably some function that it looks to be more regulated than others. Well, the thing is, we also have on the web, we have databases that are rich of previous proteomic experiments where people have studied protein co-regulation. So the principle is this: if we have hundreds and hundreds of proteomic experiments, and then we look at all the proteins that are detected, and then these proteins are always regulated in the same way. Like, for example, if in a data set, a protein is very abundant, and then this other protein is very abundant too, and in another data set, one protein is very poorly expressed, and this is poorly expressed too. Once you have hundreds of these experiments, what you can do, you can use these co-regulation features to tell, well, this protein, they must have some sort of genetic interaction because they are always regulated at the same way. And there are software available online that if you paste a set of proteins or even just one protein, they can tell you what are the other proteins that seem to have exactly the same trend across all of these proteomic experiments. One of them is called co-expression. I'm sorry, I forgot to put the citation here, but you can look for co-expression on the web. And another one is called Proteome HD. And you see here, it may be more clear the practical example. This is your query here in purple is the protein H.1. And then what the software is extracting for you is extracting all the other proteins in green that have exactly the same trend across all of these experiments, like H-1. And you see that here you have, for example, 300 different experiments. And then the software is telling you, well, if this protein is always regulated exactly like H-1, maybe it's a protein that interacts somehow, even not physically, with H-1, could be of interest for, to deconvolute the biological meaning of the proteins that you see regulated in your data set. Right, so before I go to the practical example, I want to spend just 5 minutes to a question that is, opens up, a much more complicated field, which is post-translational modification. The thing is, of course, we have quite a bit of information about protein functions. There's still quite a bit we don't know. But if we have a list of regulated proteins, we can tell somehow what are, generally the most enriched properties of this data set. But when we include as additional complication the fact that these proteins can be modified, it gets really tricky because some of these proteins have lots of modifications. This is, maybe an exceptional example. Histone proteins. Histone proteins are known to have hundreds of different modifications. This is actually getting old. There are so many more modifications that have been discovered after 2014. But each of these modifications is potentially extremely important, right? Because protein modification affects protein localization, protein lifespan, protein interactions, protein activity. And I included end identification because sometimes if you don't consider post-translational modification for proteins that have abundant ones, you might not even see them in your proteomic results if you do a search, if you're searching for spectra. But what is the problem? The problem is you have a protein, and that protein has a certain annotation, and then you have a series of modifications. Some of them might be regulated, others not. And now the question is, what is the meaning of these regulated modifications? Because all of these modifications belong to a protein that has one defined function. The answer is not easy. The thing is, there are ways to at least start approaching this issue in a non-complicated manner. So, but basically, the take-home message is that comparative proteomics, potentially can become not too complicated. But when you actually look at comparative post-translational modification, gets complex very quickly. You see, this is an example of a protein RB1 that has 1, 2, 3, 4, 7 different. In this case, there are phosphorylations, and then these phosphorylations are differentially regulated. So now the question is, what does this mean, in my data set, and what are the modifications of interest that I should consider, for example, for follow-up experiments? There's not one answer, of course, to that, but I'm going to tell you how I would approach the problem. The way I would approach it is first try to break down my experiment in smaller groups. Trying to create, for example, subcellular fractionation. These they can tell you quite a bit about what is your protein, what is your modification involved in. Here, for example, actually, let's focus on this example first. This is a typical example of the protein HDAC4, this histone deacetylase. When HDAC4 is phosphorylated, is known to be evicted from the cell nucleus. So, it's involved in regulating, in removing acetylations on the chromatin when it's unmodified. When it gets phosphorylated, it gets evicted from the nucleus. So, this is an experiment that if you had fractionated your proteome between cells that are, between proteins that are inside the nucleus or proteins that are into the cytoplasm, you can do it with a simple centrifugation. You would have found HDAC4 unmodified in the nuclear fraction and phosphorylated into the cytosolic fraction. That could help to, come to the conclusion that phosphorylated HDAC4 is evicted from the cell nucleus. When you study, here is an example, when you study, for example, synaptosomes, synaptosomes, they have these vesicles that they use them to transfer signals between neurons. If you purify these synaptosomes and you purify, for example, the soluble fraction and the membrane-associated fraction, then you have a fraction with proteins that are, for example, inside the lumen, inside the cytosol of this vesicle, and you have proteins that are bound to the membrane. And for example, you can tell if a protein gets phosphorylated, if it gets translocated to the membrane or translocated to the cytosol, for example, after a stimulation. And this can tell you quite a bit about what is the role of these red proteins, because once they are on the membrane, once they are associated to the membrane, they are the ones that get recognized by the receptor, by the synaptosome that receives this vesicle. These are not just random examples, these are real data, some of them that we are working with. Cellular fractionation really helps a lot to tell you a bit more about what is the function of a specific modification. Now, you might do, for example, an experiment where you study post-translational modification. You end up with a list of 1,000, 2,000 regulated modifications. And now the next question is, okay, can I tell what are the enzymes that are responsible for catalyzing or removing these modifications? But the answer, of course, is very complicated, but there is something we can do with at least some type of modifications. For example, phosphorylations. Phosphorylation are catalyzed by kinases, and they are removed by phosphatases. And phosphatases are more spurious, so it's a little bit more of a complicated prediction. But kinases, many of them, they have quite interesting motifs that they recognize in their protein target. So if you take, let's say, all of your regulated phosphorylations, you know from your proteomic data set on which amino acid they are of which protein, and you can check whether they have a specific enrichment on specific protein sequence motifs. Because the reason because it's interesting is because these motifs, they are sometimes recognized by specific kinases. There are software, for example, IGPS, which is a software that if you just paste your list of phosphorylations with the phosphorylation site marked somehow, they tell you what are the protein kinases that are more likely responsible to have placed all of these phosphorylations. So if you have if you are comparing a normal cell and a cancer cell, and then the cancer cell has these upregulations of phosphorylations with a specific motif, you might end up predicting what are the kinases that I need to target, that I need to drug to basically block these regulatory events. So it might easily end up being more than one kinases because kinases are not that specific. And then what you can do, for example, you can group them in families. There are software, for example, like Kinome Random, which you can use them, for example, to tell me to which families the kinases that I found belong to. And then you might use, for example, drugs that are less specific, or maybe mixture of drugs that target kinases of a specific family. And I think I'm going towards the end here. The final question that is probably more like the most important question. Once you have a regulated data set, the question is which of these modifications among these 1,000, 2,000, 3,000 that are regulated are more likely to be functional? Like if I have to put my money on a validation experiment, which kind of site I want to test, to see whether, for example, if I generate a mutant, it has a phenotype in my organism. And, question?
Senti, noi abbiamo un numero per accettare spedizioni che si chiama Eroi.
Please switch off your microphone.
I think the question is not for me.
Ricevere campioni da
Beppe, chiudi il microfono.
Actually, I am the host, so I can mute people. Give me a second. There you go. Sorry about that. Sorry about that. Right, so we're talking about functional modifications, right? So the answer, of course, is not easy, but you know, of course, people have been looking into that. For example, there is this very interesting paper that came out not long ago, and you know, these authors, they put you know, remarkable effort trying, for example, to group you know, if you find a list of regulated phosphorylation, which are the ones that are more likely to have an impact on the phenotype, you know, and then here, you know, it's obviously a little bit arbitrary how you rank phosphorylations that are more likely to have a function, but these are some of the parameters that they use to, you know, to tell which modification are probably more interesting. They look for phosphorylation events that are more conserved across organisms. They are looking for the ones that in the various data sets they had, that they show the strongest regulations when you treat the cell, or they are located at interface position. You know, there are modifications that are not, let's say, nested in the core of the protein, but maybe they are in a region that potentially can interact with other proteins. So whether they are present or not, they might contribute to create or destroy a protein-protein interaction. And then based on this, actually here they came out with the score, a very interesting score, but obviously it might not be suitable for your specific interest, and it might not be actually that you are interested in phosphorylations. It might be easily that you are working with another type of modification. So again, there's not one way of doing this, but one thing that proved to be relatively successful is, for example, to map your modifications into protein domains. And you know, in order to choose which modification is more likely to have a remarkable impact on my cell phenotype, you can start actually grouping your modification, because you know the site when you map it by mass spectrometry. You can tell it if it's within a certain active site of a protein, if it's within certain regions that is necessary for interaction, for example, a big finger, if it's, for example, within a specific motif, of which motif you know is important for something. This is just an example with protein acetylation. You can do it with any modification. And when you group proteins within motifs, you know, you have the the help of the fact that we know quite a bit about protein motifs today. So there are, so let me skip this. There are several databases now where we know, where we can map motifs based on, domains based on protein motifs. We can tell if they are specific for a specific protein function, and we can tell if they are specific for a specific protein family. This is just some of the examples, and then I also want to give you one last slide that includes a little more of the tools that I listed so far, to map and draw protein motifs, protein domains, predict whether a post-translational modification is in a region that tends to be conserved, proteins that are, databases of enzymes that catalyze these modifications. So with that, I'm going to stop here, give you just a conclusive slide, brief summary, telling you just a reminder, if needed, that mass spectrometry provides a very large amount of multidimensional data. Deconvoluting this data is, can be challenging, can be overwhelming, but it really offers a potential for generating an hypothesis in an unbiased manner. So without looking for your specific protein of interest, you can ask the data, tell me what is interesting here. And to do so, you don't need always to rely to proteomic experts. Of course, proteomic expert will do a better job, but as we discussed last time, there is a relatively simple way to transform a table into a ready to digest list of proteins. And now I'm going to show you just a practical example about how this list of proteins can be converted into a potential biological function. So let me stop here with the slide, and in 2 minutes, I'm going to show you this practical example. So we're going to open the table that we worked with last time. And then, as you remember, we basically sorted our table for this arbitrary score. This score that it was the product between the fold change and the p-value, which means that now we have our proteins that are sorted, our 5,000 plus proteins. They are sorted in a way that on the top of the table, we have proteins that are highly enriched in this RMD treatment. And on the bottom of the table, we have proteins that are highly enriched instead in the wild type or depleted in RMD. Yeah, you see, if you go to the bottom of the table shows you the ones that are most downregulated, significantly downregulated, and on the top of the table, the ones that are more significantly upregulated. So how do you convert this overwhelming list of proteins into something that you can read? I'm just going to show you a couple of tools among the ones that I already mentioned. So for example, the first one could be, we do the gene ontology. Gene ontology, we can do it with software like Gorilla. And like I mentioned, you can either set 2 unranked list of genes or a single ranked list of genes. Now, our table is already sorted, right? We want to know which functions are more specific for the RMD treatment. So our list is already ranked. So what we can do, we can just paste here the entire column containing all the accession numbers of our, our protein result. And what the software will do, we'll look at what are the functional enrichment of proteins that are at the top of our table. You can look for biological process, molecular function, cellular component, or everything. And then you just click go. You see it's as simple as that. And then the software will retrieve a list of functional enrichment. You can use the 2 unranked list, but then you need to decide at what part of the table I want to put a hard cutoff. Okay, these are the ones, this is my target set, this is my background set. It's always a risk, right? Because depending on the statistical calculation that you did, you are basically telling proteins with the p-value below this are all trash, above this are all good. It's a little bit of a risk. I prefer to use the sorting approach. Anyway, you get a list of enriched functions, and then down here, you get a list of functions that are related to which proteins are the most enriched in your table. And for example, you see the first couple are related to protein secretion. So this is already interesting because it's suggesting you that you might have done something to your cell that affects the regulation of the secretome of the proteins that are evicted outside the cell. You can look here, if you click on show genes, what kind of genes were used to generate this enrichment. So these are the list of the proteins that are found in your data set that they contribute to give a strong p-value to this enriched function. Yeah, but there is still a little bit of different things, and it might not be immediately intuitive for you about what is interesting. So what you can do, you can just take this list of genes and then paste them in a different software. So let me just isolate the gene name. You remember what we did last time? We replaced space and everything that comes after the space with nothing. And then this list of genes, for example, we can upload them in a software for protein-protein interaction. For example, string. So if you go here and you click on multiple proteins input, what you can do, you just copy-paste your list of genes, and you tell the software to let you know whether there are groups of these proteins that they seem to be all associated for a specific function. Now, we don't have time to go through about what all of these columns mean. What helps you to for your data analysis is knowing that the more interactions you have, the more confident is the interactome, and then the more these proteins are known to be associated for some reason. Right, so you can go into settings, you can work on the aesthetics, you can change the confidence of the interaction, and then update your network. And then without knowing anything about any of these proteins, you immediately notice that there is a group of proteins, all called RAB something, that are all clustered to each other, which means that in your data set there are several proteins that belong to the same family, that they seem to be all enriched in your R&D treatment. So these proteins, they actually are RAS-related proteins. They are responsible for forming, vesicles for exocytosis. So it makes a lot of sense that you found an enrichment in secretion. So that could be, an interesting follow-up. If you're wondering at the end of your proteomic experiment, what should I do next? What I would do, for example, isolate from my cells extracellular vesicles, or for example, analyze the proteins that are secreted by my cell upon the treatment to find out, okay, these proteins that are involved in transport, they seem to be so upregulated. What are they transporting? And actually, just in a few minutes, because I don't want to keep this too long, now that you know that these RAB proteins are proteins of interest for you, what you can do, you can highlight them into your final table. This is your data set, right? If you want, for example, to, like we discussed last time, if you want to plot a volcano, right, you need a fold change and a log2 converted p-value. These are the proteins that are regulated in your data set. Now, let's say that you want to show where your RAB proteins are, right? So just add an extra column here. You can, I'm just gonna call it modified p-value here. Just temporarily, I'm gonna put a negative value here, so a value that actually falls off the graph here because we have only positive values on the y-axis. And then I'm going to filter for proteins for RAB protein. So which were named, sorry, 1 second, RAS-related protein. Right, so these are all RAS-related proteins that belong to the same family. So I'm gonna filter for RAS-related proteins up here. Here we are. We have about, I don't know, 30, 40 proteins related with this name. And then you can replace this p-value with their original value right here. So now, when I remove the filters again, I end up with a table that has a new column where when I have a RAS-related protein, it has a p-value that is positive. So why did I tell you all of that? Because if you want to display this one now in your volcano, you can add an extra layer where the x-axis remains the fold change, and then the y-axis becomes the modified p-value. And then now what you have, let me just here remove, put back only positive values. Of course, you can play with the aesthetics, but what you have here, now you have a representation that tells you where are in your regulated data set your RAS-related proteins. And then you can see, these proteins are all being upregulated with my R&D treatment. So this could be a very good place to start in terms of, what is the phenotype of the cells that have been treated with this drug. And as you notice, we did all this without knowing much about the biology of any of these proteins. We just let the data guide us through a possible interpretation. I think I will stop here because obviously everything I'm telling you is, is arbitrary, is subjective. I just wanted to give you my take on how I would approach an unknown proteomic data set. But, I don't want to talk too much to affect your, your ways of interpreting data. I hope it was useful though. Yeah, thank you, Andrea, and everybody for being here. And if there is any question, of course, I'm happy to take them.
Thank you very much, Simone. Really interesting, very clear, and actually I loved it. Is there any question from the audience? You can also use the chat if you wish. Otherwise, I do have a couple of questions. Okay, I will start then.
Mm-hmm.
There's one point that is always kind of tricky to me, or at least something that I would handle with care, which is the following. While doing the gene enrichment with proteomics data, it happens that some, say, pathways are basically always activated, no matter, or sometimes no matter the experiment, no matter what you do with your system. And this is because those pathways are linked to, I don't know, a couple of thousand of proteins. So it's very, very likely that some of these proteins are up or down regulated in your experiment, and that particular pathway is reported to be dysregulated, but it might have very little meaning in your particular experiment. The other way around, you can have in your, say, enrichment list pathways that have very low p-value. Then you look closer into this data. It turns out that these pathways are represented by maybe 2 proteins. And one of these 2 proteins is highly upregulated in your data set. So the corresponding p-score is crazy low. What's your feeling about this? How would you handle this kind of outcome from gene enrichment analysis?
Yeah, no, you're absolutely right, because this problem comes from the fact that in our gene ontology annotations, we have thousands of proteins that have a certain annotation, and then we have 5 proteins that have very specific annotations, and that's because we simply don't know enough about many proteins, so we just call them. We just label them as involved in metabolism or involved in nucleic acid binding, and that's and that's a problem inevitably when you do your gene ontology enrichment, because the proteins that these proteins always show up for a reason or another, and that's why I spend so much time talking about choose the background that is ideal for you, because an enrichment is always a subset of proteins that are enriched in something compared to something else. And I think we maybe don't spend enough time discussing about what is this something else. Like I said, if we use this something else, the human proteome, we're always going to find protein, the functional enrichment of whatever model system we're working with. If we use as a background, I don't know, something that is not sufficiently comprehensive, then what will result as functionally enriched are always the proteins that are most abundant, because they will always show up in your data. So there's always going to be the spliceosome, there's always going to be cytoskeleton, and because the enrichment uses as background something that does not include enough cytoskeletal proteins. And so, I'm not saying that is the ideal way of doing it, but what is helpful is always to keep in mind that the functional enrichment that you find is always enriched compared to something, and it's always helpful to think about what is this something. And what I would do, for example, if I see functional enrichments that are very generic for me, For example, I would filter my table, let's say for protein involved in metabolic process, which is something that always shows up. I would filter my data only for proteins involved in metabolic process, and then I will relaunch the enrichment. And then now I have only metabolic proteins, but I have my data sorted for my regulated features, and within the metabolic related proteins, which ones are those on the top of the table? And that gives you a little more specificity. It's also entirely possible as well that when you do an experiment, your result is not crystal clear like that. It could be that there are groups of proteins involved in different things, but that's okay. As you saw in one of my graphs, it's perfectly normal to have different functions that are enriched, and then you can create subclusters for each of these regulated features. Then obviously ends up being your arbitrary decision which one I want to focus on first. But it's not surprising that when you treat your cells with something, or you're looking at your cancer cells, there are multiple functions that are regulated. So yeah, it's not so straightforward. But for the enrichment part, I would focus on what you compare with what, and that could simplify your interpretation.
Thank you.
Thank you, guys.